Bueno, hola, hola de nuevo. Y para continuar con el programa, it's my great pleasure to introduce you the first, the keynote speaker. This is Professor Dr. Simone Graeff-Hönninger. She's at the Institute of Crop Science, Crop Systems, and the Unit of Crop Systems and Modeling from the University of Hohenheim in Stuttgart, Germany. And yeah, she's not only a very good friend, but also a colleague, and we have cooperated in the past, and we expect to cooperate also more intensively in the future. Professor Graeff-Hönninger is an expert in cropping systems and modeling at this university, and her research focuses on cultivation and production methods for agricultural crops, including the quality of the crops. And she leads several regional, national, and international projects dealing with innovative or underutilized plant protein sources. We have had the opportunity of having her 5 or 6 years ago in the frame of the Food Security Center project, also of the University of Hohenheim. And we also contributed together to a book she edited some years back. And she also hosted one of our students in an exchange stay at her home university. And I welcome you, and the floor is yours. Thank you very much.
Well, I think now it's better. I will take you within the next roughly 40 minutes on the journey on the hidden power of cannabis odor and show you some of our results of our cannabis research. Well, first of all, let me take back a look at the history of cannabis. So looking back in history, we just know that cannabis is one of the oldest plants that was used by mankind, and it was used. We have the first documentation of its medical use can be found in the pharmacopoeia of the Chinese Emperor Shennong, which is dated back roughly 2,500 years before Christ. Cannabis was used for different purposes like sacramental use. It's known for its medical use. And you all know the use of its fibers for the textile industry or just the sailing ropes. So cannabis was intensively used then as a therapeutic drug or for these different purposes until roughly, as you can see here, the 1930s, until it got criminalized in most states due to the therapeutic or due to the psychoactive component THC. The THC, which you can see here, was then, even if it was criminalized in a couple of states in the 1930s, THC was recognized only roughly 30 years later in the middle of the 1960s. It was then isolated and politicized. Then roughly cannabis was criminalized in most of the states until the beginning of the early 90s, where there was a kind of mind change taking place. And it gained a lot of more popularity since then as a therapeutic drug. And a lot of the countries, again, allowed the cultivation of cannabis, at least of the ones that are low in THC. So we can say that now cannabis is cautiously being readmitted into the legitimacy as it raises its claim. There are multiple claims right now due to its medical benefits. And nevertheless, due to this large period where it has been criminalized, we can know that it has been largely excluded from research. And there's only a little we just know about this plant in the meantime now. And yeah, that's now where we just are at the point that there's a huge research gap and we just know only a little about this plant. So what we know, let's have a look on that. We just know that cannabis, the origin, it just lays in Central Asia. And from there, cannabis has been distributed all over the world. And as you can see here on that on these red areas, it's now been grown, and most of it can be grown more or less globally. It's grown, even if it is grown worldwide, we still have to state that its legal status varies. As you can see here, a couple of countries now have legalized, like Canada or Uruguay, and you can expect that due to the increasing demand of cannabis and due to the increasing interest in its medical purposes, this map is going to underlie constant changes Also in Germany, we're right now discussing the legalization, and it's already on its way that we just brought up the first laws that we just take the next steps into its legalization. So what else? We have to say that due to this huge medical potential, there has been more or less more claims on using cannabis, for example, for pain treatments or other treatments like epilepsy. So there's a large interest right now overall in the world to use cannabis and to use this plant for its medical purposes. But nevertheless, as research was laid back, and there is still a lot that we do not know about this plant, and there has to be a lot of research still carried out till we fully understand the plant. So talking about the plant itself, we can subdivide the plant into different organs, and out of the different organs, we have the roots, we have the stem, the seeds, and the inflorescence. And we're talking here about a multi-purpose plant because all of these different organs can be used, and that finally gains a lot more attention to that plant. For example, the roots, they can be used as a phytoremedial way, or the roots are just deal with a lot of carbon, they're able to store a lot of carbon in the soil. So when we're talking about CO2 storages, this plant has also gained a lot of interest in terms of carbon storage. The stem is classically used for fibers, and the seeds, they are famous because they're rich in omega-3 fatty acids. And out of the seeds, we just gain the proteins. And right now, plant-based proteins are another source that is really dealing for human nutrition. Nevertheless, on the side of the medical use, we are mainly talking about the inflorescence and the leaves, because these inflorescences and the leaves, they contain the phytocannabinoids and they contain the terpenes. So let me talk about a bit more about these phytocannabinoids or these phytochemicals that are contained in the flowers. Basically, the most famous one of these phytochemicals is the so-called THC, which is known for its psychoactive treatment. So these phytocannabinoids are built in the trichomes of the flowers. As you can see here, these are the little dots here that are contained in these flowers, and the flowers here are full of these little dots, these trichomes, and these little brownish bubbles, they just form up the trichomes, and within these trichomes, there we have the phytochemicals. So when talking about the phytochemicals, we just we're not only talking about phytocannabinoids, we're just talking about a lot more that is contained in these little trichomes. And as you can see here, the pathways, how these secondary metabolites are built, they are right now almost cleared in research. And we're talking, so we have cannabinoids, we have a lot of terpenes, and we have the flavonoids. But most of these compounds, out of these, only the cannabinoids have been intensively researched for the medical purposes. And right now we're just starting to dig a bit more into the terpenes and into the flavonoids in research to understand what these compounds do in addition for the medical treatment. So looking a bit more on the phytocannabinoids, one of the major or the famous ones is the so-called THC, because THC, that is known for its psychoactive compound, and basically it binds to the so-called CB1 receptor, and this is why it is basically psychoactive. It's mainly used for anti-pain, anti-nausea treatment, or anti-immune treatment, and there it is now in medical treatment. It's a lot of cancer treatments, or where opioids are nevertheless or no longer effective. Most of the treatments now are taking place with THC. On the other hand, we have the so-called CBD, which is out of the other cannabinoids that the plant contains, one of the more intensively researched ones. The CBD is not psychoactive, which is really important, because that just gives another chance to use that phytocannabinoid on a medical treatment on a different way. So now these two cannabinoids are intensively researched, but looking at their active way, we just have now understood how they just work in the human body. And basically there are a couple of cannabinoid receptors that have been identified, and they're involved with a variety of psychological processes, including pain sensation, mood, memory. So that is where these, the CB1 and the CB2 receptors, that's the way where the THC or the CBT binds. So these cannabinoid receptors are then activated by the cannabinoids, and most importantly, we have the human body has a natural endocannabinoid system. So the human body builds these cannabinoids itself. So, and now if these cannabinoids are built in the human body itself, sometimes we can also enhance that in order if we just introduce these cannabinoids into the body, and by that is clear why there's a medical treatment effect. So these receptors, looking a bit more in the detail, because we just need that later on again, if we just have a bit more into the look of the terpenes, we just I need you to understand that these receptors here, especially the CB1, where the THC binds to, and the CB2, where main THC and CBD bind to, they can be primarily found in the central nervous system, and the CB2 receptors are mainly expressed on the T cells in the immune system. So that is the basic way how these THC and CBD works when we're just talking about a medical treatment. So nevertheless, looking at the plant itself, we just know that there are not only cannabinoids, and there is not only THC, and that there is not only CBD, there are a lot more cannabinoids, as you can see here, CBC, CBT, CBG, CBN, and out of these cannabinoids, we're just right now at the starting point trying to understand what these cannabinoids do, how we could use these cannabinoids for medical treatment. And there's a lot of research that still needs to be done in order to fully understand. Nevertheless, the plant builds up all these different cannabinoids. And as you can see here, in total up to now, we have found more than 104 cannabinoids that have been identified. And probably there is a lot more in that plant that has not been recognized yet. On the other hand, we have, besides the cannabinoids, we have roughly more than 440 non-cannabinoids. And out of these, the non-cannabinoids, let's have a look on that, what the non-cannabinoids make up. So looking in the non-cannabinoids, we have to state that one major part of these non-cannabinoids make out the terpenes. And these terpenes can be either monoterpenes or sesquiterpenes. And these are the compounds that make up cannabis odor. So they're really important, not only for the smell, but right now we're at the starting point to realize what do these terpenes do on a medical point. Now, looking at cannabis odor is a bit like we just have different, depending on the strain and the genetic we use, they make up different kind of terpenes. And based on these different kind of terpenes, the cannabis plant smells differently. So it can smell, as you can see here, it can have either a piney or grassy smell, it can be citrusy, it can be woody, it can be floral, or it can be skunky. So there is also this little nasty odor where a lot of complaints are around the cannabis growing facilities, especially when it comes to this skunky odor. Now let's have a look a bit more on this, on the relevance of the terpenes. Why do they matter? So regarding on the cannabis terpenes, we can see that here there is only a slight or only a minor part here out of the cannabinoid of the terpenes that cannabis really produces. And as you can see here, now we're just digging into the part, why do these terpenes matter for medical treatment? As you can see, one of the major terpenes, for example, is humulene, we have oxymene, we have carotene, we have myrcene, and all these terpenes, they just have a different medical purpose. So some of them have a property of anti-inflammatory. Some of them have properties of, let's say, sedative. Some of them have an antioxidant potential. So depending on the terpene, it not only matters how your plant smells, it also matters what the final medical treatment out of that results. So, and so far, we're just slightly digging into that part of the terpenes. And as you can see here, what the first part, what was done in research was just to see how are these terpenes interrelated. And what we figured out was, for example, for beta-caryophyllene and here beta-humulene, there is shown that these terpenes are somehow interrelated. So if one occurs in the plant, the other one is showing up there likely too. Nevertheless, we have also the opposite example that, for example, for beta-myrcene and alpha-pinene, we don't see any correlation. So there's only a small linkage that in case beta-myrcene is exposed by a certain strain, it's not linked to any other terpenes. So there is a lot of difference that is going on by the strain we just pick, and we still are at the really, really early beginning of understanding what do these terpenes do. So nevertheless, now we have a medical plant and we want to use that plant for medical treatment. And we know that the phytochemical profile of this plant matters for the medical treatment. So now we just have to ask ourselves and say, well, hey, what influences the phytochemical profile? What influences the phytochemical concentration in cannabis? Because that is what finally matters for the medical treatment. And here, yeah, we just know that there's a lot of medical treatments that are right now investigated, but still on the side of the plant, we still are at the beginning to understand what are the components that really influence the profile and what are the components that really influence the concentration in cannabis. And that is where agriculture now kicks in, because that is definitely our part in the research that we have to contribute to investigate the plant. So let's have a look on what we know so far. And I can say, well, most of the knowledge that we just now, when you look into literature, there's a broad range of different discussions. There is a lot of blogs you find there. But basically, research on cannabis and publications on cannabis, they just started only in the last, let's say, roughly 5 years. And we just figured out that we just only know so little about that plant. So what we know is basically, we know definitely when looking at the concentration of phytocannabinoids or terpenes, we know that the strain or the cultivar definitely matters. So in terms of strains, we have multiple all over the world. There has been a lot of illegal breeding going on. So there's a lot of strains out there, but they're really roughly or badly characterized. Mainly they're characterized for their THC content, because that was the major goal for breeding, just increase the THC content, and nobody else cared about the rest. But looking now at the different strains that are out where we are going to classify the cannabis strains. I have 45 minutes or? 45 minutes. I was just confused because there was a sign. I have only 5 minutes left. So, okay. So in that case, when looking at the strains, we just classify the strains in different chemotypes. And we have the chemotype 1, which is characterized by a high THC content. And on the other side, on the opposed side, it doesn't have, or it has the rather low CBD content. We classify chemotype 2, which has a more balanced content of both THC and CBD. And we have the chemotype 3, which is normally high in CBD and low in THC. So basically, by picking the right strain, you can definitely influence, the grower can influence the content of CBD and THC. That was easy. So now, what else matters? We just know also that these little trichomes, that the size of these little trichomes, that these one really matters, because the amount of THC and all the other cannabinoids and terpenes, that is correlated to the size of these little trichomes. And as you can see here, once we compare the fiber cultivars of hemp, which are also out there, which are mainly grown for its fiber use, and we compare them with the drug strains, we just can see that the diameter and the volume of these trichomes definitely changes. And that especially in the drug strains, the diameter and the volume of these trichomes is much higher. So they contain a lot more of these phytochemicals. Okay, now that's almost a lot what we know about the plant. And what we know here is also, we just know that definitely the plant age, especially the harvest date, matters for the final concentration of THC and terpenes and CBD. As you can see here, we again have here the differentiation between the three different chemotypes. And we have here the growing time in days. And again here, just comparing THC and CBD. And as you can see here, THC content and also CBD content increases with the growing time, but it follows somehow an optimum curve. And it follows the optimum curve. Now it's a bit more flat than the others, as definitely there the THC content or the CBD content varies, but it normally follows an optimum curve. So we have to figure out when is the best harvest date. As you can see here, there we have the highest THC content, it drops again. So the growers need to figure out and just say, hey, when is the best harvest time? When should I harvest my plant? And to make it a bit more complicated, this harvest time and the plant age really is strain specific. So each grower has to figure out what is the optimum growing time for my strain that I can harvest the highest content in CBD and THC. The same holds true for the terpenes. So looking at the terpenes, we can see here that there is regarding the monoterpenes, that there is an increase in the monoterpenes with plant age, but there is a dip in the sesquiterpenes, and there is so the grower here also when he looks on the terpene side, he also there has to take into account that the plant age matters. And we just here have, as you can see, that there is an interplay between these terpenes and the cannabinoids. We definitely, for final medical product, have to figure out what is the optimum harvest date. So now, that was all what is so far known about that plant. There's a lot more that is not known. So, and therefore, there is a lot of research that is now needed on, and that's also carried out, on the cultivation system, on fertilization, irrigation, on pruning, on plant density, on light. So all these economic factors, we just do not know really, or there's only roughly hints in literature, but they're somehow contradicting depending on the growing system, depending on the strain. So it's really hard to figure out what do these really, the list here contributes, and how does this matter in terms of the terpenes and the cannabinoids. Now, I want to show you now in the next couple of slides some of our research we did on these couple of questions here. And let's start with the question of harvest time and pruning technique. And we just here investigated in the study the impact of having harvest time and pruning technique on plant biomass, on the CBD, and the total CBD yield of a chemotype 3. What we did was we implied here three different pruning techniques, one which we called here lollipop. As you can see here, the one that's a plant where we just exclude, or we just took away all the side branches. We had the one called topped, where we just topped the plant at the 10th node, and we have the control plant, which was not pruned at all. And our question was here, as you can see, that the topping definitely has an impact on the amount of flowers and on the distribution of the flowers on the plant. The question was here, how does this really impact the cannabinoids and the terpenes? And looking at the results, we could say that the pruning technique itself, it didn't matter really for the total CBD content. It mattered for the total CBD yield of a plant because it had an impact on total plant yield. But the CBD concentration in the flowers was not affected by the pruning technique. Nevertheless, what we saw here, as you can see here, we just also subdivided the plant in three different areas, where we just had a more closer look on the three different flower types, where the flowers on the top, in the mid, or in the low. And what we can see here was that the inflorescence position, that this was definitely significantly affected by, and that the CBD content really changed depending on the flower position. So we just know now, okay, depending on where we pick the flower, and finally, if you hand over your flower to the patient, it really matters where you take the flower from. So now just figuring out that the inflorescence position matters, we also had a closer look on the terpenes and not only on the cannabinoids. And here we just did that study for 3 different strains. And we just subdivided these different strains into the main top bud, which is the highest top bud level. We had the side top buds, and we just subdivided them into the side buds and just analyzed all these different fractions a bit more in detail. And we just here, that is just one of, or just a couple of the terpenes we had a look on. We just had a look on the more than 20 of the terpenes, on just to getting a bit more of the full profile. But I only show you here 3 of these. And what we saw here, as you can see, these are the 3 different strains. And what we can see here is that basically we figured out that there is a heterogeneous distribution of the terpenes that has the chosen interaction with the morphology of the plant and the flower position. And we figured out that the more shaded the flowers are, the lower the monoterpene concentration. So definitely we have to take care of the flower position, and we have to take care of what are we going to harvest and what do we finally hand over to our patients. Now, looking on the other side, on the other question of how does fertilization, does this also have an impact? Here we just tested 2 different fertilizer types, mineral and organic, because there's always the question of, can we grow cannabis on an organic part? And we applied the fertilizers in 3 different dilutions, mainly diluting the nitrogen. And we again investigated the impact on biomass, CBD yield, and the nutrient use efficiency. And what we just saw here, first of all, here's the inflorescent dry matter. We just saw, okay, definitely over time, the inflorescence dry matter just increases. So we took here a couple of time harvests. So that's logical. I think that is something you can expect, that over the time that inflorescence yields goes up. And we could see here, okay, it also changes depending on the amount of fertilizer we provide to the plant. But on the other side, we could see that regarding the CBD yield, that there is no more significant change between the 160 and the 240 milligram per liter nitrogen. So we definitely figured out, okay, that there is a certain limit in terms of nitrogen fertilization, and that there is a recommendation that the plant in that case would not need more than 160 milligram of nitrogen. So looking then at the terpenes again, nevertheless, it gets a bit more complicated. When we always look in the terpene, it gets a bit bumpy, and they go up and down, and they just do whatever they want. And so what you can see here is that in terms of alpha-pinene, we just saw that there is a slight increase. But for limonene, we just saw, okay, there is a decrease, then there is an increase again. And for linalool, there is a slight constant decrease. So definitely fertilization matters, and it matters on what kind of terpene we look at. It also matters on what kind of cannabinoid we look at. So we just have to state here that the fertilizer amount definitely impacts the flower yield. It impacts cannabinoid and terpene contents. And depending on the kind of terpene we look at, it increases or decreases. So with that, based on the fertilizer you provide to your plant, you can change the terpene profile. You just can, or at least the terpene amount. You can definitely, with that, also have an impact on the odor of cannabis. And nevertheless, as the odor changes, we come back to the question, how does this relate back to the medical application? Because if you remember the slides I showed you on the medical way that these cannabinoids work, we can assume that definitely it matters for the final application. So now, looking at one other major factor, I think everybody knows that light definitely matters for the plant. And it's the same that holds true for cannabis. Cannabis is one of the plants that really attracts high light intensities. It has its photosynthesis, it just goes up, and it's just fascinating on that part. But looking at not only on the light intensity, but also on the wavelength, we just know that there is an impact of wavelengths on cannabis secondary metabolites. As the wavelength they just, the different wavelengths interact with the different photoreceptors. So now the question is, under which conditions, under which light conditions do you really grow your plant? Because with that, you can definitely impact the phytochemicals. So looking a bit closer on the light, we just carried out a study, again, picked 3 different strains. Out of these strains, we just knew that these strains definitely differ in their terpene profile. And here we just definitely wanted to know, what does the light do to the terpenes? As you can see here, we had one strain that was really dominant in alpha-pinene. We had another one that was a bit more of evenly distributed, and the one here that was dominated by alpha-pinene and myrcene. So these 3 strains, we just picked them and we just exposed them to 3 different light regimes. We just picked here 2 of the LED fixtures, which is the Solray and the AP67, and another one that is the ceramic haloid, metal haloid lamps that are the standard lamps that are basically put up in every greenhouse. So looking at the light distribution and the wavelength distribution, we just knew, okay, these all these different they have a different light distribution, and they also differ in their red-far red ratio. So now, what did this do to the cannabis growth and to the, especially also to the phytocannabinoids? So looking at the phytocannabinoids, here you can see that there are the three different fixtures, or the 3 different light sources. We again split that up into the 3 different strains. And here I show you the CBDA content. In the plants, the lowercase letters always indicate the comparison between the strains within a light spectra. The uppercase letter indicates the comparison between the light spectra within a strain. So let's have a look because that here is the light spectra, how the light spectra matters. That is what is of interest to us. And what we can see here regarding, for example, this one of the strains here, the Canada, we just could see, okay, there is not much difference depending, this strain is not going to react. But we could see that the light, especially the LED fixtures, always led to a higher concentration of CBD. Nevertheless, looking at the E19 strain, we saw that there is also a significant impact of the light source on the amount of CBD. And the E19 strain definitely brought up more cannabinoids under the ceramic haloid lamp than under the LED fixtures. So there's a slight hint there that definitely the source of light matters in terms of influencing the cannabinoid profile and the cannabinoid contents. So let's look on the terpenes. And again, I picked here the alpha-pinene and the carotene as an example of 2 of the terpenes. Again, you have the same way of how the figures are represented. And what we can see here is regarding the alpha-pinene and the carotene, first of all, okay, we just see that there are differences, and the terpenes again react differently to the different light sources. And what we can see is that the light source here, for example, if we just again have a look on the E19 and the carotene, we just see, okay, there is change here. And again, depending on the light source, and again here for Canada, there are significant changes depending in the terpene profile and the terpene content, depending on what kind of light source we pick. So we definitely have to state that the light source changes the terpene concentration. And depending on this, depends on the terpene and it depends on the strain. And this is of high relevance for the growers and definitely for the medical application, because we can turn the plant upside down by just picking a different light source. And that is, we're just trying to raise the awareness to the growers that that really matters, because as the terpenes interact with the final medical application, you need to keep an eye on what you're doing here, and you need to keep an eye on the growing conditions. So now, and then we just looked a bit more deeper into that and just did a biplot analysis. And what we could state here was, again, there are a couple of terpenes that are, as I've just shown you also on the previous slide, that a couple of the terpenes are not interrelated, but each of the strain has its unique terpene profile. What we could also see was that there is the terpene concentration is positively correlated with the main top bud flower yield, but it's negatively correlated with the side bud yields. So in case we have an increasing side bud yield, there is a dilution effect on the terpenes. So basically, we can conclude on the results on the light studies that the secondary metabolism in cannabis can be artificially manipulated by the choice of the light spectrum, the light source, and also the intensity. And this finally has an impact on the medical potencies. Okay, now looking at the medical potencies, because that is where we just finally go for with that product. And looking at how that final product ends up with the patient, it takes its way. So there's the grower is going to harvest the bud then goes to the pharmacy, and the pharmacy just sells it to the patient. So the patient comes there with a recipe from his doctor. He comes to the pharmacy, and the pharmacy just gives him the bud he wants, or just he was prescribed to. So basically, these buds or the flowers on the prescription, the flowers are right now only screened and classified for the THC and CBD content. So the patient gets a recipe, and the recipe states you are prescribed a flower that contains 10% of THC. We do not know anything else about the other components because only the flowers are classified for THC and CBD. Nothing else is known about the flower. So then we have to ask now, just as I showed you, how we just can turn the plant upside down in the growing process. We just have to ask how to standardize the bud in the cultivation process. Or we have to ask the question a bit more critically, should the bud be used as a medical product at all? And we're not only going to sell the bud, we're also using full spectrum extracts, so that there's an extract made out of the bud, and depending on the terpene profile and the rest of flavonoids and all the others, it definitely has a different impact on and the bud can change, even if you just use the same strain, but the bud can change depending on the growing conditions. And then we just also have to clarify what phytochemicals we didn't take so far into account and what finally make up the medical effect. So now what we did in order to get a bit more in that detail, we just took the full spectrum extracts out of our flowers, and we just worked together here with a company. And this company, they provide cell models to test the full spectrum cannabis extracts. And they tested these especially for the CBD receptors. And the CBD receptor is the so-called TRPV1 receptor that is found in the nervous tissue. It's involved in feeling of the pain. Yeah, and this is more interestingly also activated by the capsaicin, which you can find in chili peppers. So now this here, we focused here in that study on the TRP1 receptor that is mainly activated by the CBD. It's not reacting to any of the THC. So in that case, the THC content of the samples didn't matter. Now, when we just apply CBD as CBD as a standard, first of all, to these cell models, what you can see here is we have a fluorescent signal that caused an increase of the intercellular calcium that is triggered by opening this TRP1 receptor due to the impact of CBD. So here we have 0.0 before the application of CBD. Then we applied the CBD, and we can see here the calcium channels open, and there is a change in the fluorescent signal due to the impact of CBD. Now, looking at what we do, that was the standard of CBD. Now we just applied our full spectrum extract of different strains. And we just picked here a sample of 2 plants out of the same strain because we just wanted to see how the replicates just work there. We just picked here a CBD content of a strain that was roughly around 6%. And what we could see here, that was our strain E18, 1 and 2. So same plant, grown in the same conditions, but there were slight standard variations, but nevertheless, the reaction within that strain due to its CBD content was the same. There's a bit more variation here. And then we just also had a look into the terpene profile. And as it was the same strain, the terpene profile was almost similar, just expressing beta-caryophyllene and alpha-pinene. but they just differed here. There were slightly higher contents in alpha-pinene, where we just assume that this causes a bit more of that standard deviation. Now we have a strain, so we just can say, okay, well, there's a similar reaction if we just apply a full spectrum cannabis extract and it does what it should do. Now, on the other hand, imagine that the patient comes to the pharmacy and he gets a CBD strain that is prescribed with 6% of CBD. Then the pharmacy, they have different strains available and they just can hand over whatever. It's not necessarily specific that there is a specific strain ordered. The patient gets a strain with 6% CBD. So now we just picked here two different strains, the Canada and the E18, and again, applied them as a full spectrum extract to the cell tests. So now here again, here we can see that there's the reaction that the cells are opening, calcium just flows into the cell. But on the other hand, what we can see here for the other strain is we just have the opposite reaction. So there is an inhibition of the cell models or of the receptors. And now we just were asking, say, hey, but the CBD is 6%. So why do we just get a total different reaction here? And when we now look a bit more closer on the terpene profile, as you can see here, the red one just corresponds to that one here, and the green one corresponds to that one. So we can see that the terpene profiles are totally different for the two different strains. And these terpene profiles definitely lead to a full different reaction in the cell model. So you can expect that there's a different medical treatment, or that this is what also patients report, that depending on when they go to the pharmacy on one and they just get a certain strain, then it works fine. And on the other hand, when they come back 4 weeks later, they just get a different strain, but with the same CBD content or the same THC content. It just leads to fully different medical application here. And that is, when we just look a bit more into these terpenes, then we just see here that especially the beta-carotene that has an anti-inflammatory, and here the myrcene or the oxymene, they have a sedative, anti-convulsing, or pain-killing effect. So in that case, we definitely have to state the terpenes matter, and we should be more sensitive to the terpenes, and we definitely have to take them into account. So let me conclude on all that. I'm almost in time. That we have to state that cannabis is a natural product, and we have to keep that in mind. Large amounts of the cannabis secondary metabolites have not been investigated yet, and there is a lot of research that still needs to be carried out to fully understand the relevance. And besides the strain itself, the cultivation system really matters, and we have to be careful on the cultivation system because it definitely affects the secondary metabolites and thus the medical potency. The consumers finally need a standardized product, and here it's the task of the research that has to be carried out in order to standardize the cultivation of cannabis. And I think that is the way or the road that we have ahead of us, that there's a lot more of research that we will see in the next couple of years that needs to be carried out. And no matter what, if you just like the odor of cannabis or not, and we definitely have to conclude that there is a hidden power in its odor, and we have to take care of that. So with that, let me thank you for your attention. Thanks to my group of PhD students and postdocs, and also to the ministry for funding our projects. Thanks.
Are common floral odors, aren't they? Linalool and beta-pinene, alpha-pinene, they're pretty common in the plant world.
Yeah, definitely. They are pretty common, as I showed, as I introduced. You just have them in citrus plants. You have them also in other plants. But it's always the combination here that we just, in case if we are using a full spectrum extract, that we just see that they just combine, that there is a combination of the cannabinoids and the terpenes. There was just a recent paper published also that tested each single terpene in combination with the THC. And what they just showed was that the reaction of the THC and how it binds to the cell receptors is modulated by the way, by the terpene. And they just did, that was a really nice study, just published 4 weeks ago. And they really picked that out for each single terpene. And I think it's the combination, it's not the terpene itself, it's definitely the combination as it modulates the binding to the receptors.
Thank you.
Hi, I was just here for the conclusions, so I apologize for that. But I do understand what your point is with the combination of the different compounds. So my background is from agricultural business instead of chemistry. And I'd like to know how this would apply in the means for regulations for hemp, for example, meaning that I wouldn't be interested in using those combinations or cannabinoids. I'd rather use the grain instead. So from your perspective, do you think it'd be possible to help those farmers trying to get the grain with those added costs for the tests and the compliance? That would be my question. Thanks.
I mean, we have to differentiate. If we just use cannabis or if you use the hemp ones, the hemp strains for the seeds and for the oil or the protein, we're talking about different strains. What farmers in Germany and also in Europe right now do is that they're trying to gain both. So they just try to harvest the seeds, and they're just trying to extract out of the leftovers, out of the leaves, and also the some of the seed leftovers. They're just trying to extract a couple of these cannabinoids in order to get an extra money income. Nevertheless, we have to come up that the contents of cannabinoids and terpenes are quite low in these industrial hemp cultivars. They're roughly about 1 or 2%. So the question there is always, is it from an economical standpoint, okay, you have the leftover, it's there, but can you really compete with a strain that has 20% of CBD, or did I get your question wrong?
Great, thanks. And for the environment here, the latitudes that we are located within 30 degrees from the equator.
Cannabis grows everywhere. It's a weed. It just grows. You can just grow everywhere.
And that cannabinoid, THC specifically, does it act to help the plant adapt to these tropical latitudes? Does it give protection against fungi and other biodiversity?
I mean, regarding the biodiversity, hemp just attracts a lot of pollinators. So in that case, you can just grow it everywhere. And it doesn't matter if you just pick the tropical ones or if you pick whatever other region, it just grows everywhere. And regarding the biodiversity, that really, it just enhances, and it is one of the plants that is really discussed intensively for enhancing the biodiversity.
We also have the coffee break right now, so maybe you can interact in the coffee break. I'm very sorry for interrupting you, but it's the sake of the time schedule. So well, thank you very much, Simone, again, for your very interesting talk, for your active discussion, and we will meet back here.